Hello everyone. Welcome to the offline event of Swarma Club tonight. My name is Fan Yang. I will be the host for tonight. And it's heavily raining outside, and thank you so much for coming here in person today. For everyone who are joining us online, thank you for your support. Before we actually start, briefly introduce what Swarma Club or Swarma Research Center is. It is a non-profit scientific organization focused on complex science and artificial intelligence. It is founded by Professor Zhang Jiang. The community members of Swarma Research Center come from research institutions, universities, and private enterprises here in China and around the globe. The focus of this institution ranges extensively from many interdisciplinary subjects, such as mathematical science, computational neuroscience, physics, psychology, artificial intelligence, network science, and etc. Now, I'm very honored to introduce tonight's speaker, Professor Konrad Körding. His research focuses on computational neuroscience, concentrating how brain functions through data science. And his early research concentrated on perception and movement. And in recent years, he has expanded his work from data science to explore various fields such as still neuroscience and also personalized deep learning, and even like new directions like reverse engineering and the entire nervous system. And additionally, he's also a major advocate for open science and one of the founders of NeuroMatch, which I believe many of us has been benefited from when we have no clue how to explore fields like deep learning or computational neuroscience systematically. NeuroMatch hosts a series of courses such as computational neuroscience, climate science, deep learning, neuro AI. We all benefited from it because of him. So let's get into the topic. Welcome, Professor Körding.
Can you all hear me? Well, in this room, you can probably hear me. I hope the stream can also hear me. Otherwise, someone will let me know. Thanks so much for having me here. It's so random that I'm here. Yesterday, I gave a talk at PKU, or no, the day before yesterday. And that's when I got invited to join you tonight here. And it's a great honor to talk with so many wonderful computational neuroscientists. I will talk about how to obtain causality in neuroscience and generally talk about the logic of how we find out about the causality between items. Let me briefly start out why I'm doing it. Why am I here? Why do I talk about computational neuroscience? Why do I even care about it? And this is just as a reminder, the reason why we do neuroscience is usually twofold. We want to cure diseases. Now, if we could make Alzheimer's, if we could make all these different brain diseases go away, this would be a major achievement for mankind. And the other one is we want to understand brains and arguably ourselves. And there's a big difference in how we do those. And I think the first thing is always, let's be clear about what our questions are. And to understand brains, different neuroscientists mean very different things with what they mean when they say, let us understand brains. And so philosophers have for a long time asked what scientists, for example, like when I started my PhD, I was interested in how we can recognize an object and separate it from the rest of the world. I was interested in binding, in attention. Why was I interested in that? Because I believe that we need to understand human intelligence, or it's at least very helpful to understand human intelligence if we want to build artificial intelligence. And that was my goal at that point of time. So, but let's talk about the different kinds of questions that people ask. There's a set of evolutionary questions that people ask, which is, here we have a brain of a human being, or why does it look the way it looks? And it turns out that we share an evolutionary history with very simple things like fruit flies or even little worms. And the key piece is that we take the brain, we divide it into pieces, and then we mature each piece so that it ultimately becomes a brain like ours. And there are a lot of neuroscientists working on such evolutionary questions. And I should say, this is Tinbergen made a list of different questions that biological scientists ask. David Marr made another list. And then I think that there's some new things that no one yet talks about. So I want to first go through the 4 questions of Tinbergen, and I'm going to relate that to David Marr. So the second class of questions that people ask are developmental questions. We are born into this world knowing very little. We see all these things happen around us. We live in this arbitrary world with all these things. And in the end, all the experiences that we have shape us and give us the brain that we have right now. And all the thoughts that you have right now are being driven by the developmental process, by all the things you learned in your life. There's another way of thinking, which is goal-directedness. You could say, this is something that I did a lot of research on. How do people know how far away an object is from them? Well, they can use multiple things. They can use the texture on it, and they can use the disparity, the angle between the eyes, to estimate how far away something is. Now, in the world in which humans work, it's important to be as precise as possible. Now, you don't want to drop your coffee. And because you don't want to drop your coffee, and because the niche of humans has this property, we should expect that people optimally combine what the eye angle says and what the texture says. And indeed, there's a lot of experiments that show things like that. That is goal-directed thinking. And then there's mechanism-directed thinking. Now, like, how do I figure out how far that is? Well, there's this thing in the outside world, and there's a couple of neurons active in my retina, and those neurons in the retina go to the neurons back there, and ultimately there's a cascade of neural activities that make me recognize how far away it is. Now, these 4 questions are very different, and it's essential to know which of these 4 arguments a paper wants to make. And they always confuse one another. For example, if someone writes an evolutionary argument, we should have something that's good at detecting things that comes at us. The reason being that we live in a world where like stuff is being thrown at us or something. If someone makes an evolutionary argument that doesn't say anything about development, there's lots of ways how we can make the adult beings that are successful in this world. It doesn't say anything about the goal. It's just like an evolutionary argument. It's like, because we were very simple beings at some point of time, we still have the residuals to it. It doesn't say anything about mechanism. There's lots of ways how evolution could have been doing it. Similarly, if someone talks about the goal, like be as precise as possible, walk from A to B with as little energy as possible. It doesn't say how the brain does it. No, like there's billions of neurons in the brain. They can do it in all kinds of different ways. And when you see neuroscientists disagree, they often don't actually disagree. It's just that some of them like this argument, some of them like this argument, some of them like this argument. So the fact that there's different questions means that you always want to know which of them it is. A lot of neuroscientists believe that mechanism is the only right way of thinking. Hope that I'll somewhat convince you that that's a bad idea. Now, Tinbergen was a theoretician of biology in the 60s. David Marr was a theoretician of neuroscience 20 years later. Unfortunately, it doesn't look like David Marr actually read Tinbergen, which is a little unfortunate. He says there are 3 levels. There's the level of the goal. There's the level of the algorithm. It's a little unclear what they mean with it. And then there's the level of the mechanism. Those 2 are shared. Marr ignores evolution and development. How do we become the animals that we are? How does our nervous system as we have it right now, how does that come about? And then Pojo, who was a collaborator of David Marr, tried to add that 20 years later. Now, all those questions are different to the questions a lot of machine learning people ask. Like machine learning people might ask about the brain, can I predict as well as possible the next word coming out of Konrad's mouth? Does that relate to evolution? No. Does that relate to development? No. Does that relate to a goal? No. Does that relate to mechanism? No. It doesn't relate to any of these things. In fact, it's a little unclear what the question is that go with it. And yet neuroscientists use it all the time. And then they try to get the answer to the machine learning problem and try and answer evolution, development, goal, and mechanism based on it. Levels of indirection in computational neuroscience almost never work because we're indirect, the answers that we give to questions are then often no longer good answers. So let's do a little bit of a tour through neuroscience. Neuroscience is undergoing a truly interesting revolution that I think will change absolutely everything. So let's talk about old ideas that used to be good ideas that no longer are good ideas. There's the old idea that the brain is in a way simple. For example, when I see something, stuff goes into my retina. From there, it goes to the LGN. It's a part of it basically sees bright versus dark pixels, if you want. From there, it goes to simple cells. Simple cells tell us, is there a line here like this? Is there a line like this? Is there a line here? Is there a line here? And then it takes all those cells, according to the Huber-Wiesel theory, and puts them together to produce complex cells. And they get the Nobel Prize, where they said, for their discoveries concerning information processing in the visual system. Only that their studies didn't at all explain how information is processed. They asked how it's represented. They stuck an electrode there. They said, like, look, the neuron is always active when there's something here. Did they show how the neuron figures that out? No, they did not. Or for complex cells, the neuron will always be active if there's something in this area with roughly this orientation. Did they show how complex cells work? No, up to this day, we don't know how they work. All they did is they made a statement about representation. And the thing that made it feel as if they were producing an understanding of mechanism was that that people believed that the brain was just so simple. Like it goes from the eye to the retina, they do one thing, every cell does the same thing, and then it goes from the retina to the cortex, and every neuron does the same thing, and you keep iterating through it. That sense of simplicity is absolutely not working. For example, here's some visual experiment where they showed that in that visual experiment, if you also play tones, the tones change what happens in visual cortex. And not only the tones change, the way you move change, and the reward you get changes, and the task that you have changes. So the old notion of how neuroscience works, which is assume it's simple and figure out in which way it is simple, is dying. And why is it dying? It's dying because of engineering. Like we can now record from 10,000 neurons at the same time. We can show them stimuli, natural scene stimuli that are really complicated. And what we're progressively finding out is that there's no simplicity of any meaningful kind there. Here's something closer to what my research was on motor cortex. Georgopoulos is finding like, no, motor cortex does all kinds of things. And here's a finding from our own lab, from Ramkumar's work, where say reward is really, really important. In fact, so much so that reward describes about as much variance in primary visual cortex as movement, which is very worrying if we think about it, that movement cortex is just like this piece that like a marionette moves our arms. Like it does really interesting, complicated things. Here's another thing that I did, which is if you want to know how neurons causally interact with one another. We have three neurons, A, B, and C. Neuron A excites neuron B excites neuron C. And of course, if we look at the correlations, A is correlated with B is correlated with C. A is also correlated with C because A indirectly through B influences C. And then we developed, and this is work from the lab from 2008, great algorithms that pull that out. Or here we have a different, in the lower diagram here, we have a slightly different setting. Again, the algorithms I developed work great. Now let's think a little bit through what I did here. So I assume there's three neurons and only three neurons. They get no input from anywhere else in the brain, and they don't affect anything else in the brain. And then my algorithm works just fine. Now, primary cortex has about 1 million neurons per cubic millimeter. My cortex in the brain, I think, must be rough. My visual cortex must be like roughly this size. So it's much bigger than a cubic millimeter. Many, many, many millions of neurons. And one neuron has on average 10,000 inputs. So that setting is entirely unrealistic. And yet, me and lots of people in the field applied these methods called functional connectivity methods to neuroscience data and then pretended that the results the algorithm gives us basically tells us how neurons causally interact with one another. But that is fundamentally misguided. Like it is based on an assumption of simplicity that reality just doesn't give. And it therefore affects all kinds of answers. If we don't have simplicity anymore, evolution happens on lots of channels at the same time. Development, every neuron might learn differently as we go through our life. The goal might have like lots of dimensions to it, and so on and so forth. So that's one part. There's the old idea, the brain is simple. People needed to make that assumption because they had almost no data. But that way of thinking about the brain was wrong. Now, do we have a new way of doing science? Well, I don't know, but we certainly have new experiments. So this is an analysis where you have, by Stevenson, we call it Stevenson's law. On the x-axis, you have the publication of a paper. On the y-axis, you have the number of neurons that they were simultaneously recording from. And what you can see is that the number of simultaneously recorded neurons faster than exponentially increases. So on average, doubles every 6 years. It's clearly faster over the last few years. Why is it faster? Because we have so much great engineering. Like Elon Musk is bringing company, Paradromics is a company in that space. Lots of different companies bring development. Now, in principle, this should scale just like Moore's law because we make this out of semiconductors. But it has traditionally doubled every 6 years, and the speed of your computer doubled every 2 years. So what gives? Well, because there wasn't that level of professionalism. So clearly this is happening, and it's amazing. We also have that whole molecular revolution. We can now select a type of neuron and specifically optogenetically activate just that type of neuron. So we can now do perturbation studies. And we also now have all those data coming from post-tracking. Like as you see me online here, you can take my movement and convert it into skeletal tracking with very high precision, knowing exactly how I move my arms through 3D space. And just to mention a project from my own lab, we've been working with the Michael Platt lab to basically have enclosements with lots of cameras tracking monkeys, even using data from lots of species, training big foundation type models on that, and then fine-tune for domain specific questions. For example, here in primate face, we took 65 genera of monkeys, including great apes, and all kinds of lemurs, tarsiers, all kinds of cool monkeys. Now, why do we train with all these different monkeys? Well, from foundation models, we know that if we take text from lots of people, lots of languages, Chinese, English, all the languages together, we do better than if we just train on one language. And in the same way, if we train on lots of monkeys, we do better than just training on one type of monkey. Including on humans, we can do better by also training on monkeys if we have enough data from all the monkeys. Now, like here you see the two sides. We have the ways of thinking about brains that are all going downhill. And we have all these experimental techniques that are unbelievable in how they scale. So what are the new things that we could have? And I think there's one thing holding us back, which is the focus on mechanism. Now, I told you there's 8 kinds of questions on the first slide. But neuroscientists are uniquely focused on only mechanism. So for example, NIH very specific says that what they want to fund is understand fundamental mechanisms of development, structure, and function of the nervous system. The NSF, these two are the big American funding agencies. The NSF wants to fund innovative proposals aimed at advancing a rigorous understanding of the neural mechanisms of human cognition. It's uniquely mechanism. Now, what is mechanism? Now, if mechanism is the one thing that is fundable in neuroscience, so like that's what people often say. We only want to fund mechanisms. It's not mechanisms on neuroscience. But what is mechanism? Well, it sounds like a great word, mechanism. We all agree mechanism sounds great. Now, like, and arguably what mechanism means is if we say A mechanistically affects B, we mean two things. The first thing is there must be a causal chain on A. If we would go in and do something to A, something would happen to B. And an understanding of this chain in terms of lower level component, like we have two neurons, A and B. I want to understand A makes B fire, and I want to understand it in terms of the component, which is by virtue of having a synapse that connects A to B. Now, causality, like that means that if we want to establish mechanism, we really need to establish this. Otherwise, it's not mechanism. Otherwise, it's maybe some fun finding, but it's not mechanism. Let's look at what causality is, not like the relationship between cause and effect. The best definition that I know is by this cartoon by XKCD. I used to think correlation implied causation. Then I took a statistics class. Now I don't. Sounds like the class helped. Well, maybe. The reason why this is a great definition is if you learn about causality, you learn to realize that because there's a correlation, I took the class and now I don't, doesn't mean that it's the class that did it. Maybe like it would have happened at that point of time in my life in any case. So it's the ultimate definition, that's right. Now let's make this a little formal. Let A and B be events. Causation exists if and only if we had changed A to A star, the probability of B would have been different. It's a very simple intuition. Now, I don't know about you, but I knew that correlation isn't causation. Now, they taught me when I was a first-year PhD student in physics. And yet, intuitively, I always felt like whenever I think about something, like I go home, there's pie, probably someone baked it for me. Like it's kind of like it seems like the kind of things in my life. Correlation is causation. If stuff happens, then it happens for a reason. I observe the reason, I observe the thing. And where's it coming from? Like, why is it that simultaneously everyone teaches us that correlation isn't causation? And yet, if we think about it, correlation is causation. And in fact, if I talk with neuroscientists, it's always that experimentalists that find a correlation, like they stick electrodes, they find if this goes up, then this goes up. They will 100% of the time ascribe causality to it. They will ascribe mechanism to it. Where's that coming from? So I want to go with you through a very simple mathematical system to build these intuitions. Now, like this is the simplest possible dynamical system that you could have. We have xt, which is a vector that is the state of the dynamics that we have. We have xt plus 1 is some nonlinearity times axt plus epsilon t, where this is an n-dimensional vector. We have a sigmoid nonlinearity in front of it. We can entirely take it out. It's just so that like the system doesn't become unstable. A is the real causal matrix. Now it says which thing affects which other thing at the next point of time. You can think about this x vector as just a set of activities of neurons. Not like maybe we have 6 or we have 100 of them, something like that. And then we add random noise, we initialize to 0, we set the largest singular value to 0.99 to make the system stable. And then what do we expect? Now, if like maybe neuron 1 strongly positively affects neuron 2, then I expect that I often have neuron 1 very active at one time step, and then neuron 2 be very active at the next time step. And so it's the delay 1 correlation that should be similar to causality. And then we chose n to be 6, and we simulated it. This is what we have here. We have a 6 by 6 system, so we have a state space that is 6-dimensional. On the top, we have the true causal matrix, how every neuron influences every other neuron. On the bottom, you have the delay 1 correlations. And look how similar they are. Wherever there's red, no, like red, there's red on the other one. Okay, so in this 6-dimensional dynamical system, causality and correlation are very, very similar to one another. So does that mean that correlation is actually causation? Actually, no. So here we do the same dynamical system, only we go from six dimensional state to a 100-dimensional state. Nothing else changes. So like the whole equations that I gave you is exactly the same. And now those two seem very, very different from one another. And of course, we can quantify that. Look, if we have a small system, the kind of small systems that Konrad can simulate in his head. No, I can think of 3 variables, like you talk to her, she talks to him, and like he says something. Like it's a kind of system that I can simulate. In that case, there's a very high correlation. Like we just take the matrices, flatten them to standard correlation, and we find very high correlation for small systems, and we find very low correlations for large systems, and it happens just outside of the kind of things that we can simulate in our brains. So that's why we go through this world with that mistaken belief that the correlations that we actually observe are causal, but they're not. And we can make that be much more mathematical. So this is the famous omitted variable bias equation. It's very popular in economics. Let's say we have a variable, we want to estimate the influence of x, a vector, on y, a scalar product. Then we have that y is x beta. So those are the interactions between the variables that we do observe. And then we have other variables z that we don't observe that also matter. Now, like I record from 3 neurons, there's also the 1 million other neurons that we have locally. Each of them with their causal influence plus noise. We can then, the way we estimate that is regression. This is the standard equation for regression. If we put that through, if we just put this in here, we get that the expected value of the causal effects as we estimate with it is the true one, which is good, plus, and then we have here these extra bias terms. And now look how this term here has the same form as this term. So that means that the variables that we don't record, they produce random biases, each of which should be roughly of the same size of those that we do record. Now let's think a bit about it a little bit. In that cubic millimeter, we might have like 3 neurons that we record from and 1 million that we don't. That means that we have 1 million or 999,997 neurons that we don't observe that like should be producing biases for us. Now they're all of the same size. So the real signals that we have will be completely drowned out by the signals that we don't have. And therefore, for those neural signals, we shouldn't expect to be able to get a connectivity between things. This is particularly important because there's a large branch of neuroscience called functional connectivity analysis. It's very popular in EEG. It's very popular in fMRI. There's thousands of funded studies in this area that basically use this approach, ignoring the many, many dimensions that they don't observe. And that's, of course, a problem. So here's kind of the intuitions that we have there. Now, we are in the age of Google. We have so much data. Why can't we make the problem go away? Here's the logic of the problem going away. We have variables A and we have variables B. And we have so-called confounders. So a confounder is a variable that affects A and also affects B. For example, it could make one neuron active at one time step and then the other neuron active at the second time step. It will look as if the neuron makes the other neuron active one time step later. And confounders make it so that it looks like 2 variables are correlated, even if there's no causal relationship between them. So the standard assumption, standard idea that machine learning people tell you is like, well, then let's correct for them. And in fact, economists and lots of people working in causal inference are trying to remove the confounders. And that's a good strategy. The problem is there's also colliders. So a collider is a variable that is affected by A and B. Think, for example, what are the colliders for neurons, for 2 neurons in the brain? Well, all neurons in the brain, because if I have 1 neuron, if I wait a little bit, it will affect every other neuron, which means that the activity of every neuron basically tells you about ultimately what happened with all the other neurons. So every neuron is a confounder for every pair of neurons, and every neuron is a collider between a pair of neurons. If we correct for a confounder or our correlation gets to be closer to causality. If we correct for a collider, it gets to be further away from it. So even if you gave me unlimited data, like record everything, like, or take healthcare, like we want to know if a treatment works. It would be great if we didn't need to run a study. The problem is we never know what's a collider and what's a confounder, and almost everything is simultaneously collider and confounder. Therefore, we can't correct for it. So that is the cause of causal inference. Now, you can say, well, do advanced data analysis methods help? If you look at modern neuroscience, people use UMAP and they use non-negative matrix factorization. They use lots of different techniques. And in fact, if you look at, say, uses of machine learning, it's been growing exponentially. I should redo that graph at some point of time. It's getting much larger. So it's people use these modern methods, but will they help? Well, no. So they're useful for specific patterns. And here's kind of what people do with supervised machine learning in a sense. So like they use it for engineering where it's perfectly fine. Like I want to have a prosthetic device, someone lost their arm, I want to use the brain to control a prosthetic device. All I want the prosthetic device to do what the person wants to do. I don't care if it's like, if it knows how the brain works, I just need it to do what the person wants to do. Machine learning, no problem. Logically perfectly fine. Lots of people use it for identifying predictive variables. You give me some neurons you record from, and you're asking me, Konrad, does visual inputs matter for them? Do auditory inputs matter? And like usually the answer is yes. And machine learning is good. You can also use it to identify diseases with it, for example. You can also use it logically to give you a benchmark for another model. Maybe you come to me, you're like, I have this model of like how the brain works, and I'm like, okay, machine learning describes 80% of the data, and your model just describes 3% of the data, and therefore like kind of like maybe your model misses something important. You can also use it as a model for the brain. That's a completely different talk. I very strongly promote for brains being in lots of ways similar to deep learning systems. I should also, there's cases where this is the only way of doing this. For example, let's say you want to know during my sleep, how do I move through space? Well, it's very difficult. You can't ask me like, hey, Konrad, wake up. Where are you in like your world? That will never get you very far. But you could say, if while you're awake, I can build a supervised learning algorithm that tells you where you're in space, and then I can apply it at night. And then I'm like, oh, look, you're like very quickly walking through this little maze while you dream. I should also say people are like really pushing like supervised learning in that area. This is one of my favorite papers. Eva Dyer, she was a postdoc with me a while ago. So they do basically train a big foundation model for it. They don't take data from one animal, they take data from all the animals they can get, brain data, movement data. And then they build like a really cool adaptive system that kind of fits foundation model and ultimately does much better than anything else for neural decoding. Does that tell us how the brain works? No. Does it tell us what the mechanism is? No. Does it tell us what the goal of the brain is? No. Does it tell us how the brain developed? No. Does it tell us how the brain evolved? No. But it's really good at solving supervised learning problems. So the key here is that these advanced techniques are not a shortcut to tell us about the question that we really want to ask. Here's a fun visualization of that. No, people often use dimensionality reduction techniques. They take the brain data, they know like it's now 10,000 neurons at a time. Let's just do dimensionality reduction so that we have far fewer dimensions. Now, will these dimensionality reduction techniques reveal a real structure in the data? No, because they see structure. So if it was a Gaussian and we apply PCA, now this is an example with PCA. If it was a Gaussian, then telling me the two axes of it describes all the data as well as possible. But if it was a mixture of two things, it describes something that doesn't exist. Now, this dimension is not in the data, and neither is this dimension. There's these two dimensions in the data. But the dimensionality reduction algorithm doesn't tell us. And in this case, it tells us dimensions, but it doesn't at all describe what's really there. So it tells us things that aren't there, and it doesn't tell us things that are there. So therefore, it's very difficult to make progress at understanding brains by putting them through dimensional correlation. So this is all just to say, the modern techniques that we currently have haven't yet given us an alternative to what we originally wanted to do. We have real legitimate questions about the 4 questions. And none of these modern techniques get us there. None of the big experimental data sets, they don't really get us there. It's very hard to even imagine how we're going to get there. And this is going to be the problem that you will have to solve during your career in the field, and that I hope the field will be solving over the next couple of decades. But let's take for granted the assertion that neuroscientists make that mechanism really matters. So let's talk about how we could get at mechanism. So this is the worm C. elegans. It's arguably the most boring thing that you can think of. Now, like, it's a little worm, lives in the ground. What does it do? It goes around, slithers around, and eats bacteria. Now, C. elegans can do almost everything that we can do, like minus talking, if you're malicious about it. You know what do I mean about it? It goes and finds bacteria, which it then eats. It finds other worms that it then copulates with. It lays eggs into areas where it's a good idea to be laying those eggs at. It can find, if it's too hot, it finds places that are colder. If it's too cold, it finds places that are warmer. If there exists some bacteria that are noxious to it, that hurt it, it avoids those bacteria. It can learn which things to seek and which things to avoid. So it has a very rich repertoire. The cool thing about it, it has only 300 neurons. Because it only has 300 neurons, oh yeah, and it's also transparent. Now, like, this is like a microscopy image from it. So because it's transparent, we can see all 300 neurons at the same time. And experimentally, the field has established that it can measure the calcium concentration, a marker for how active the neurons are, in all 300 neurons at the same time. So we see all neurons at the same time. We can also perturb every neuron. We can go in there with the laser and be like, these 2 neurons zap, and then those 2 neurons will be active. And then we can see how that changes everything that happens in that one. So if we want to do mechanism neuroscience, I think we have to start at something simple. Because if we're honest, what we do with big systems is we do some measurements and then we hypothesize what the mechanism could be. But these hypotheses, quite honestly, I've done a lot of computational neuroscience, a lot of modeling in my work. These hypotheses that we have, they're very brittle. So causality, we can do imaging where we see all of them. We can then perturb all of them. We can then, now what's a neuron in that view? A neuron is something that gets inputs from other neurons and produces an output that affects other neurons. Now, we can call that the computational neuron primitive, which is that a neuron's effect on the rest of the brain is through its one output. Now, if a neuron's output is very high dimensional, then this approach will fail. But if we can say a neuron's output is a function of its input, and only the output affects other neurons, then we should be able to simulate it with it and like put that back into behavior and see, does that simulated system now work like a real system? And I should just say, you do need to stimulate, in principle, if you record everything, then the confounding problem goes away, not because all confounders are observed, but that doesn't really help you, because if you don't stimulate the system, it like sloshes around in a low-dimensional manifold, and you cannot invert a system if you only have low-dimensional data on it. So this is just from our simulation. It suggests that we need a lot. How are we doing on time? Yeah, let's talk a little bit about quasi-experiments. So what I told you so far, it's very hard to have causal information about something. Why? Because it's hard to have the power and it's hard to identify the system, because of all the confounders. So there are cases, though, where we can do that. So imagine what we want to know is what's the causal influence of X on Y. And of course, we have confounders, like the Z that we don't observe. It's just stuff happening in the world. Confounders are always there, and the Z's affect the X and the Y. And then at first, you will say, well, if we have X and Y, we can't figure out how X influences Y because there's confounding. But now imagine, and this is a quasi-experimental technique where you say there's a so-called instrument. So that instrument is a random variable that only affects the treatment. It affects all the rest through the treatment. Now you could say this could be for a neuron. I have a variable and it makes the neuron fire or makes the neuron not fire. And this variable is random. So the idea here, like let's just briefly zoom out. The idea behind such quasi-experiment instrumental variables is that there's sometimes cases where if you want the universe randomizes things for you. For example, you could be in a situation where the threshold, where you're being close to the threshold of a neuron is ultimately a random variable when something happens. And now in that case, so let's be clear what the assumptions are. The important thing is that this instrument cannot be affected by any of the confounders. It must be something where something happens randomly. And you can also say maybe it must not directly affect the outcome. If it directly affects the outcome variables, everything is going to go down. Now, these two assumptions, you can never test them in data. But you can be in situations where you can be relatively positive about it. Let me give you examples. So like instrumental variables, maybe your doctor is randomly chosen. And some doctors like prescribing one drug and other doctors like prescribing another thing. Maybe you grew up at a random place in Beijing and you're forced to go, this is an American thing, you're forced to go to the nearest school to you. In that case, your house might be a little over and then you'd be in the other school. And it's like totally random because people there maybe don't buy their houses based on where they are relative to that. In which case the instrument is where exactly your house is. So there's a lot of cases that will have that structure in the world. And now you can say, well, does that help? Well, yes. So if you have the so-called instrumental variable, the thing that is like actually random in the universe and that we observe, our estimate is going to be unbiased. If you assume that things are independent, which they're not, like it's like if you ignore the confounding, you will be biased. But I want you also to see, while you're biased, you're much more precise. And this is the general strategy when you do causal inference. What you often do is to get better from a causal perspective. You're willing to be worse at how good your predictions are. You're willing to accept more variance for becoming unbiased. Now, keep in mind that I told you it has these assumptions, like exclusion, restriction, IV must not affect the outcome. It must not be affected by confounders. What happens if that assumption is wrong? Well, then it's both very noisy and very biased. And in fact, it's more biased than if you don't do that. I should say there's lots of different quasi-experiments, and we built a fun little tutorial for anyone being interested in that. And you can get an overview of possible causal inference techniques there. Now, without further ado, let's talk about a case where we can solve causal inference in a very different way. So in supervised learning, much of what I do is neural network supervised learning. You know what the inputs are, and you must have ground truth data. Now, you must have a data set that tells you what's right. Now, like if you have machine learning for predicting diseases, you tell me this is the movements of a person. Yes, they have Alzheimer's disease. This is the movement of a person. No, they're healthy. And so on and so forth. You need ground truth data. That's the idea of supervised learning. Now, where do causal inference algorithms come from? Like I told you about instrumental variables. Instrumental variables came from a clever economist saying like, yeah, like if these things are really independent in this and this and this way, and not independent in this way, this is how we could figure out how causality works. So the way it works is we have one expert, very bright, knows lots of things about the domain, who designs an algorithm, and then we take the algorithm and we use it on the data, and it tells us this thing causally affects this thing or doesn't causally affect this thing. Now, this is one way of how we could do it. But keep in mind, different experts come up with different answers for that. Now, like some people talk about instrumental variables and regression discontinuity design and deep learning and inverse variance weighing. There's lots and lots and lots of algorithms in the causal inference field. And in fact, if anyone is interested in causal inference, I can highly recommend the books of Julia the book of Julia Pearl, and the book of Why is a great introduction to the field. I can also highly recommend a very different book, Mastering Metrics, which comes more from an econ perspective. So, but like, let's do it differently. Like humans, so who knows what the bitter lesson is? Okay, so bitter lesson is the single most important observation about deep learning, which is whenever there's problems for estimation, it goes through multiple stages. At a first stage, there is a problem and there's no solution. Then a clever human being comes and builds in all kinds of things. Think about speech recognition. Like in the beginning, they started with something that models the human cochlea and that models pieces of the brain. It's like and models lots of like human knowledge about speech. And time passes, more data came in, computers get faster, and now speech recognition is just we throw all the data that we have at it. It's a transformer architecture. There's nothing about it that is speech specific. It's just a vanilla transformer, and it outperforms what the humans did before. So if we zoom out from that, the bitter lessons, like every new problem is first solved really well by a clever machine learning domain expert. Time passes, and then a general purpose learner just outperforms what they did. Why is that? The human who designs it builds in lots of knowledge about it. And the knowledge is kind of right, but it's not quite right. No, in reality, no variable isn't affected by anything else. So everything is kind of confounded somewhere. When it comes to speech, like, sure, the face isn't quite there, but it turns out that people use face for low tones. And so on and so forth. There's always, people can build in things, but they're always only half right. And if there's enough data, like then these half rights that the humans introduced is ultimately a liability. So that's the bitter lesson, which is as time passes, anything a data scientist like me does eventually becomes irrelevant because the general purpose learner will outperform us. So let's do that same idea for causal inference. Let's say we don't have one causal inference problem where we want to say, here's a data set, tell me if this causes that, but give me 1,000 of them and give me truth in all of those cases. No? How can we obtain truth? Well, we can like zap the neurons, or we can like go and give someone a pill when we're in medicine. We can do all those things. And so we don't take one problem, but we take 1,000 prompts. And then instead of the human making the algorithm, we're like, okay, give us the algorithm that works best on the 1,000 things that we already have. So we convert our problem into a learning problem, into a general purpose learning problem. And then the algorithm that is learned from lots of past cases can then predict what causality is. So it turns out that causality is one of these areas where there's really missing ground truth data, and producing good causal ground truth data would be a massive service to the field. So we started with something where we can know causality, which is my favorite system. This is the MOS 6502 microprocessor. The great thing is we have a great causal simulation of that. And then we can say, can we figure out the pieces of the causal simulation based on data that we have there? So what we ask is, we have a transistor, and we can put extra current into that transistor. We're asking, does this transistor affect the voltage that goes into another transistor? And there are cases where we have a causal thing. So here we have one transistor that causally affects the other transistor. And we have the temporal traces. Like we go through time. Like we just had the microprocessor basically play Pong. And then we have non-causal. And we have thousands of them. We basically tell the algorithm, well, no, these two, this one actually didn't influence the other one, well, this one did. If we look at this a little, what do we see? We often find that this one transistor goes down, and then the other one transistor goes down a little later. It's not always happening. It's more complicated than that. And how do we get at ground truth? Well, we just do experiments on this. We basically put some extra current in, we see what happens with it. And we do honest validation, which means we use half of the data to train our causality detector, and we use the other half to see if it actually works. And then standard machine learning. Now, like the whole idea is that we're going all in on like standard machine learning. Standard transform architecture, pairwise sequences into an embedding layer, sequence encoder, global pooling head, tells us the probability that the connections are causal. And it works great. So here we have LSTM, TCN, transformers, modern neural network architecture. Here we have the things that the brilliant statisticians came up with. Just correlation, which is just ignoring the causal problem, mutual information, Granger causality, all popular methods that neuroscientists use quite a lot. And what we see is the machine learned causality estimators are really good. The human ones are really not so good. Also, like the machine learning ones, of course, can deal with noise because we use that for learning. And the human ones cannot deal with noise. And we can say, well, does it generalize? Yes, we train on one of the games and then we test on the other games. Works just fine. And we can do that idea called GradCam, where you can ask a neural network, which information are you using? What it's doing is it really focuses in on the times where there's fast transients of things, and then uses the information from you. So what does this all mean here? No, the cons is it requires a lot of ground truth data. It's not that we ask one person. And it's also, the results are entirely impossible for humans to understand. No, it's a transformer, it has 1 billion parameters. Okay, our transformer doesn't have 1 billion parameters because no one gives me the money to like train 1 billion parameters, but it has plenty. It's impossible for a human to understand. It's also biased. It's going to use all the information they can, including the whole idea of machine learning is that you want to have the right kind of biases. But the pros is not like, if we will have enough ground truth data, if we have enough causal path data points, we will outperform all humans. Why? Because like their assumptions aren't quite right, and the data gives us exactly the assumptions that are really right. There's actually meaningful proofs for that, and the proofs are inherited from empirical risk minimization. The basic idea is, if you've gotten it right the last 900 times, you will probably do it okay the next 100 times. And out of that, you can get meaningful, probably approximately correct proofs. It's also a new approach to get into messy observational spaces. Now, take-home message of the whole talk is just a reminder, neuroscience is deeply about mechanism. Mechanism is about causality. Causality is something we can't have usually, which is the big tragedy of neuroscience. But neuroscience is going through this massive revolution where we have so much more data. It's like, I mean, like everything you do, everyone in this room does in neuroscience is going to feel like magic to people from 10 years ago. It's just like, there's all those opportunities. It's unbelievable. And causality is still hard. The new techniques don't deliver us causality. Like, yes, there's optogenetics, but optogenetics is low dimensional. Sure, I can hit the system, see what happens. But the system has, every neuron has 10,000 inputs, 10,000 to one output. I can't hit all of the 10,000. In fact, I can usually only hit one in one experiment. And I should also say, I think there's new ideas from economics, like instrumental variables, and ideas from machine learning, and I hope that they all come together to help us actually solve mechanism problems. I'm not saying I have the solutions for that, but I feel that as a field, we will solve that. With that, thank you.
Thank you.
Oh, no. The model only gets correlational information. So basically, the only reason why it works, no, I mean, like, this is a very deep, wonderful question. So how can the algorithm do it? Because everything is confounded. No, like, basically, only say why the clock is running does the microprocessor do anything. Like, there are all those, there's all kinds of confounding factors. The reason why it does is you confound it and you collide it, but you can say, from enough data, you can learn about the structure of confounding, and you can learn about the structure of collider. And therefore, you can do these estimations despite the fact that there's just locally, it's an impossible task. Now, like, take medicine. Take socioeconomic status. Now, like, socioeconomic status, rich people get more treatments, do better in the end. But at the same time, if you get treatments, you become poor. And if you're in the process of dying, you also become poor, at least in America. So it's a collider and it's a confounder at the same time. But keep in mind, if I give you a really big data set, it can learn to correct for those things. It can be like, yeah, it's a little bit of a confounder and only a tiny bit of a collider. And therefore, let's like 90% correct for it and ignore the 10%. And it can kind of, effectively it can learn really clever strategies that are not locally possible. It can basically have a model for the pieces of the confounders that are modelable and the pieces of the confounders of the colliders that are avoidable. And basically in that space, then it can do well. Yeah, so let's be clear about the logic of that. There's nothing about the algorithm itself that would convince me that it gets at causality. If you would write down the equations for me and I'd look over the equations, I'd be like, I have no idea why this would be correct. But at the same time, it comes with mathematical proofs. What's the mathematical proof? It says the last 1,000 pairs of transistors that it's seen, it's gotten 90% right. Therefore, in the next 100, I should expect that it gets 90 right, and it certainly won't get less than 80 right. Now, that's the logic that is behind it. So it's now why that works. Now, like it basically, it's in that space where from a pure causal inference perspective, it is not identifiable. It just becomes identifiable through specific aspects in the data. Now, like it can, for example, use timing information really well. But timing itself, A can come before B. But it can discover that, like, basically confounding in timing is pretty weak, and then correct for that. And so that's the weird thing. It's kind of breaking out of the standard logic that correlation isn't causation. It's not. But it corrects and it exploits any aspect of the data that you have here. Okay, so this is a great question because the field of causal inference for people who enter it looks really bizarre from the outside. What I mean with that is there are about 5 different communities working in that area, and they all say that all the other 4 communities are entirely useless. Now, the Book of Why by Judea Pearl is very much Judea Pearlian. So there's one group of people, mostly in statistics, that do causal inference and that very much are focused on the way Judea Pearl thinks about it. The other book I recommend, which is Mastering Metrics, and it's Angrist and who's the other? Pishaska, I think. Oh, yes, exactly. Josh Angrist, Nobel Prize laureate. The field, so they approach it very differently. So Judea Pearl's school very much focus on mathematical proofs of under which circumstances systems are identifiable. Josh Angrist and that general econ school of causality very much focuses on thinking through the set of possible confounders. So they're much more experienced at thinking, okay, what could be a confounder? So at some level, Judea Pearl assumes, his school assumes that we know what the structure of the world is. The economists have like a culture of asking, well, I don't think that they're not confounded. What about age? What about like this confounding based on where people live is where other people with similar political opinions live? They have a rich literature of thinking in this direction. That's why I usually recommend to people to hear Judea Pearl's side, which is a great introduction to the causal inference field. The only one thing you want to ignore is that Judea Pearl invented absolutely anything in causality. And instead, the economists have very complementary strengths, and social scientists, where they focus on well, what could be confounders? How does the world really look like? So they have very different ways of thinking. And that's why I think those 2 books are like wonderfully complementary to one another. Yeah, this is a wonderful question. Like, what's software, what's hardware? So you can say neurons feel like hardware. Like they're like wires. They're like wires and amplifiers and all those things. At the same time, well, what is software in the brain? Well, it's wires. So the connection between 2 neurons, which is a hardware structure, is what carries your memories. Like it's the synaptic weights and synaptic properties and calcium concentration and so forth. That like it's the calcium channels that carry this. So basically, the software is hardware, but also that hardware is like it's all coupled with with one another. Also, what people don't usually know is neurons have synapses between them. They have these dendrites. But what they also constantly do is they try and make new connections. They're like, okay, bad connection, let's try something else. They change their connections all the time. So even the wiring between them, it's not that we have the wiring and then we just put in the weights. It's even that wiring, what's connected to what, is changing all the time. So from that sense, like the thing is that in the brain, there's only hardware, like there's not really software. And that level of abstraction where we can say in computers, hardware and software are strictly different is not true in brains. But it's also starting to not be true in the circuits we use to deal with artificial neural networks. And like we use FPGAs, where it's like not that there's a memory bank with the weights and then there's like the processor. Like it's kind of, we're trying to, in modern hardware design, to bring the relevant bits of the model very close to the compute that we have there. So the answer is basically in brains, the cut between hardware and software is very complicated. And progressively, even in artificial neural networks, the cut between hardware and software is getting weaker. Yeah, so this is great that you're homing in on that. Now, like this is a violation of the computational single neuron doctrine. You can say a gap junction is an output that is not the somatic output. So now, how can one deal with it? If we built, now this is it's a great look. Like I think like this pushing on where the pragmatic assumptions are. That is the main thing that we should all be doing in neuroscience. So if we're to understand brains, we have this long history of like basically lying to ourselves that we understand so much. And in this case, what is it that one can do about them? So the first thing is, if the gap junction transmits effectively what the soma of a neuron does, then this is not very pragmatic. Like it's still just like this one-dimensional signal, maybe with like a delay effectively in it. But as long as neurons are isopotential, it's not a problem. To the level that the neurons are not, it is a problem. And then you can say, what is the next thing we can do? Well, we can then fit like all neurons at the same time and hope that we can somehow do a more black boxy model of what the neurons are. Or even if we can model where they are, and we have basically cable equation models for what every individual neuron does, then we can try and solve all of them at the same time while also fitting to the data. But like, it's wonderful. You're putting the finger exactly on where it hurts, which is to the level that this idea, a neuron has its influence on other neurons through its one output. To the level that this goes wrong, then a lot of approaches in neuroscience fail. And if we talk about gap junctions, they aren't unique to C. elegans. Like your brain has plenty of gap junctions if you look in the right places. Well, the reason why we choose C. elegans is to identify causality, you need to be able to perturb. If you see only a small piece of a system, you will always be confounded. So if you want to get at mechanism, you need to see everything and you need to be able to stimulate everything. And that's where C. elegans is just so wonderful, because A, it's small enough where you can see and stimulate everything, and B, it's highly stereotypical. Like two C. elegans worms are like in first order approximation similar to one. Okay, so this is no. So what we usually do when we think about recurrent systems in the neural network literature is that we think about a temporal unrolling. So we have a copy of the neural network at every given point of time. It requires us to have sufficient temporal resolution. C. elegans is nice because much of it is relatively slow there. So once you have it unrolled like that, then every neuron only basically every stimulation, every observation just lives at one time point. And in that sense, the fact that it's recurrent isn't overly concerning. So that's the best thing we can do at the moment. Wonderful. Yeah, so imagine you have a machine learning system that's good at making dynamics predictions. Then that system doesn't need to be causally correct. So it could, for example, be that if I then go in and change what a neuron does, it fails at that. Let me give you an example. ChatGPT is really good at answering the questions that I have already answered in my life and I've written about because it's in the training set. If I would change anything in the context, so I go from the data set that I trained on to an outside data set, I will fail miserably. In fact, like, can easily fool ChatGPT or like whatever the Chinese equivalent of large language models is by just by asking questions that are significantly outside of the training set that it has. So if you want to get out of the domain the generalization, you will generally want to get the causality right. And now let's take the dynamics prediction versus the causal inference in it. So in practice, the dynamics in most situations where you record, which is a small number of situations, they lie on like somewhat low dimensional manifolds. They like move in a low dimensional space. Now, if as soon as you give new behaviors, it forces it out of that space. And so therefore, even if you're very good at predicting the dynamics, and machine learning, deep learning is the best to predict the dynamics of neurons. No, it's the best that we have, but it doesn't need to be causally right at all. No? So like what it does, it says if neuron 17 is active at some point of time, neuron 18 is active at the next point of time. But that doesn't mean that number 17 makes neuron 18 active. So it's the distinction between what happens and what would happen if I would go in and would do a change in it. And that distinguishes correlational from causal questions, like prediction. And it turns out that it's much easier. So if you want to solve the causal problems, you almost always have matrix inversions in it, at least when you do it from observational data. Whereas if you make predictions, you don't have it to that same level. And that's why basically you can just regularize. Not like machine learning works because we regularize so strongly. And that doesn't matter because if we cut away dimensions that just don't help us so much predict the dimensions, but those are the dimensions where causal things happen. And so you need to get all those dimensions right for the causal problem, and you don't need to get them right for the prediction. Great question. Okay, I'm not sure I understand the link to causality. I mean, like just the background to make sure everyone's on the same page. The brain is often argued to be critical. So if you're in two very similar situations, the future of the dynamics may still be very, very different. It's often being implied as that's the result of self-organized criticality, that there's mechanisms that kind of make it, that similar stimuli can, or similar brain states can lead to very different outcomes. If we analyze the microprocessor, that's definitely not self-organized. We also find that it's critical. So it's I don't think the argument quite holds. But can you make the argument again to like, now how does criticality relate to causality? So I mean, like you're making the point for causality being necessary, you know, like everything is correlated because of criticality reasons, if you want. And because that is, but that doesn't mean that like neurons are all causally influencing, though we know that they're not. So I mean, like in that sense, it's like, but I'm not quite sure about the logic of, not like if I have two starting points of the brain and they diverge exponentially from one another, how does like it would seem that it wouldn't like really make causal inference harder, no? Now, like the thing is, like without perturbation in either system, I will be having a very hard time to figure out causality because unless I observe the whole system. And if I do observe the whole system, the question is, do I like really cover that whole system? Like the whole system has an exponential sized state space, and because it's exponential sized, I can't like really cover that with stimuli in a way.
Discussion, and this is a wonderful opportunity for communication. And this is obviously and we have Professor Körding from university, Professor Bing from
Hey, long time no see.
Professor Bing from this group of strong scientists. Of this event, and welcome everyone. And this Everyone give a round of applause for our next section. Our first topic will be challenges and prospects of causal inference and computational neuroscience. And let's start now. Welcome.
So can I use both, or I just use this one?
You just need that.
Yeah, I think I just needed this one. Okay. So I think the question is challenges and perspectives of causal inference and computational neuroscience. And to be honest, I see it full of challenges. You see the questions I proposed, and frankly, I think the causal inferences in the brain is, and for me, it's really like a mess, because first it's like which animal model that's what we're using. If we're using like a very complicated models like human, maybe we can only get the data from fMRI. Then I think the fMRI data is a little bit how to say, the spatial and temporal resolution are not so high. Yeah, maybe we can still get some information, but from I think we can still get some useful information from fMRI data for the causal inference, right?
So I think I disagree with you on that. So,
Okay.
So what we get from MRI results is, like, to be clear, fMRI results are very slow. Like they give you one sample a second. So
Yeah, exactly.
if you're lucky. And because they're so slow, you don't have a mechanism to find out which direction information flows. Now, you can't do this temporal unrolling because you're basically only, like it takes a neuron a few milliseconds to affect another millisecond. So that means it has the chance to go like 100 iterations through the brain before you get the new sample. And because it has so much time, basically time is useless.
Yeah.
And then you can say, well, is space useful? Well, like space is hopelessly confounded because we record in an MRI, we record at once per second from maybe 30,000 voxels, depending on how far we put push the limits. But we have 30,000 voxels and we have 10 to the 10 neurons. So we basically record from nothing.
Yeah.
So what I'd say is we can't even get weak information about causality. I'm saying that MRI has nothing to contribute to learn about how causality works in the brain, just to be provocative for the discussion.
So, okay, next time when I get a review paper using causal inference, using the fMRI data, maybe I should reject that. No, I'm kidding. So if I think, even if we take the C. elegans, the simplistic neural system that we can we use as an animal model so far, because by the way, we're also working on the C. elegans system. We made some perhaps the most detailed C. elegans models in the field.
It's incredibly cool work.
Yeah, thanks. For us, it's when they really look very closely at C. elegans. We found this really matters. That I said the weird connection between three neurons with gap that might exist in C. elegans, but mixed with chemical synapses. So that's even make your causal inference harder because it's a mixture of gap junctions and chemical synapses.
That's right. And I should mention one more thing. It's also extracellular neuroproteins, all kinds of neuromodulators.
So volume transmission. Yeah, the volume transmission. The volume transmission.
And that's people. So basically, neurons in C. elegans often communicate by just throwing chemicals outside of the cells, which then somehow make their way to the other neurons and have an effect there. And people, when they work on cortex, always pretend that it doesn't exist. And then you look at the mouse, and it has even a lot more than C. elegans in that space. So all the things that are complicated on C. elegans will be 5 times as complicated in the mouse.
Yeah. So I look forward to your data. Yeah.
Okay. So thank you for giving such a great talk. And my question is about the your experiment results. And I remember that you show that the transformer have outperformed the traditional method, like information-based method. So you believe that the transformer can do some causal discovery work. So
Indeed.
yeah. But as we know that the causal discovery have something to do with the causal graph structure. So like the V structure embedded in the graph structure.
Right.
So how to intuitively explain why the transformer can do such kind of causal discovery?
This is a very complicated question.
Yeah.
So look, like part of it is clever cheating. Let's talk about clever cheating. You want to figure out what the light switch over there is. How do you do that? No, it's a causal inference problem. You will probably tell me if I would turn that, then probably the light in that part of the room would go off. How can you do that? Well, you have flipped lots of switches, and you know that like light switches tend to be close to the lights that it switches, and you know that things that look like light switches often like switch lights. And so you can say that there's clever cheating here, because it's not that you figure it out by like doing real causal inference by like switching it and seeing what happens, but it's by utilizing kind of additional information, namely that it looks like a switch and so forth. The same thing is possible with transistors that go up and down. Like you can say, well, actually, like relationships between transistors, there's those in the IMU and those in other places, and it's kind of using a lot of additional information that we traditionally don't think of as being causal inference. And so that's how I interpret that. I know you can say, well, but that's not causal inference what I mean with it. But what do we mean with causal inference? Well, I just want to get like the graph out of the data that I have. And if it's good, then I'm happy with it. If it like uses information that I didn't think would be information, then I'm very happy with it. Now you could, so for example, say it could know implicitly that transistors that go up and down really, really frequently are often connected to other transistors that go up and down really frequently. And that is not like something that mutual information or anything like that would pick out. And you can say it's not like traditionally even causal inference. It's not looking at like conditional responses of neurons to other neurons. It's just kind of using all the information that's there.
Okay, so within the transformer, we know that there are some internal structure called attention matrix. Is this attention matrix has something to do with the causal matrix shown in your formula?
So in other domains, we try to look at that a little bit, and extracting the structure of the world out of transformer architecture seems extremely difficult.
Yes.
To the level that I'm a little bit, so there is a whole subfield of machine learning that's called interpretable machine learning or explainable machine learning. And I don't believe that's really possible, because basically, if you make your system so simple that a human can understand it, they're no longer deep enough to actually solve real-world problems. Well, that is my hunch there. Of course, like who knows? I might be proven wrong next year, but there's some statistical reason for why kind of pulling out those pieces of information might be very hard.
Okay, thank you.
So, thank you for the talk. It's really interesting. And actually, my background is neuroscience, and but not that computation. But yeah. So for myself, actually, unfortunately, the whole neuroscience area, almost we are, investigating the correlation, not causality. Actually, it's always like this. So, but for from my side, actually, I think for C. elegans, I think it's I didn't I didn't work on that, but from my understanding that it's still really hard to predict the behavior, even you know all the neurons and you know all the neuron firings, everything. But actually, it's really hard to predict the behavior of C. elegans. So, will the causal whatever computation will help on this, or is there any further benefit from that to giving more process prediction? And the first question. A second question is about is that so? I think C. elegans is also very complicated to investigate. Is there anything you are thinking about? Maybe even simple than this? I don't know, but yeah, because it's really hard to predict anything like that. Behavior. So yeah.
So that's a great question. Maybe actually I would like to give that to Professor Du first.
I just share some personal view. When I started to model the C. elegans, one of the things that I learned is the randomness, because I forget this question to you. How can you correlate the causality and the randomness? Because one of the reasons that we do not understand how it behaves is the C. elegans, they can like randomly to give the output. And it appears that when you have this small, even if you have this very small size network, it can automatically, it's like an emergent randomness. It can automatically generate the random output, even if you give some constant input to the network. So this is also my question. How can you reconcile the randomness of a network and the causality?
Right. So in principle, I see no trouble with randomness. Now, you could say causality is perfectly fine with randomness. Like if A causally affects B, if it changes the probability of B, if I change A to A star. So like at some level, that isn't really the thing that makes me not sleep at night. The thing that does make me not sleep at night is that we don't know. So one of them is how can we average across many animals? And like clearly, if we want to figure out a simulation of C. elegans, we can't do one experiment. We will need to, in our power calculations, do 10,000 experiments, 10,000 animals. But now those 10,000 animals will be different. Now, like you and me are different, like to C. elegans are different. The question is then, how well can we carry that over? The other thing that really keeps me up is how many parameters will we need? Now, will we need 1,000 parameters, in which case 10,000 animals will probably easily be enough? Do we need 1,000,000 parameters, in which case we might really be in trouble here? And so I think those are the areas that are difficult. Now, a little bit, like it's an inverse problem that we'd want to solve there. It's we observe activities and we can change activities, but what we want to know is how does one neuron affect another, or how does the many inputs of a neuron affect the output of a neuron? And for that, ultimately, we will need to make sure that we cover the relevant space. No one knows how complicated the C. elegans neuron is, although you probably thought more about that than most people. But if it's complicated, if it's relatively easy, like the neurons are nonlinear, but they're not super nonlinear, then we can realistically solve that. In principle, if every neuron can calculate any function, including the n-bit parity function, we have very strong proofs that we can that any identifications NP have space. So we kind of have to build on the assumption that there's some level of simplicity in the system. Yes. And in fact, like anyone doing any kind of science has to build on those. So like if we want to interpret motor cortex data, it requires us equally to make those assumptions. Even if we just want to build decoding devices, we need to assume that like there's a sense of simplicity in the code.
Yeah. I think let me just add one more comment, because the reviewer also asked us some question about our C. elegans model, and they said there is a fundamental question in both neuroscience and especially in computational neuroscience, is how detailed how many parameters are enough. So why we need like we need 10,000 or 1,000 parameters to build? Can we just use point neural models or some more abstract neural models? Then what can the very detailed C elegance model use for? So our answer is like, at least we think we can build a model we can call the ground truth model. You can make abstraction, yes, but at least we can have something close to realistic nerve system. So I think this, and then these two opinions, they do not like conflict with each other. They can.
Right, and you can make even more points. So, for example, there's another cool new technique where we can use expansion microscopy, where we can take an animal like C. elegans, 1 millimeter long, and make it basically like this big, make it a few centimeters big. And at that level, we can see where the molecules are. So you could say, if we are at the detailed model, the kind of models that you work on, you can say, if I say to you, well, there's like so and so many sodium channels here, it tells you something about your model. So it's a deeper bridge to the biology. Now you can say there's two ways how we could model it. Like detailed cable equation models with the molecules and everything we have. And the advantage is we can use all the bio-knowledge that we have. The alternative is that we just throw like black box models at it, where we're like, okay, let's have like a 300-dimensional state space model that we like throw the whole transformer business at it. And that might be great at predicting behavior. It might in fact be better at predicting behavior than, say, cable equations, but it will be harder for us to interpret. And the cool thing about these cable equation models is they're very interpretable.
Yes, exactly. So there is one feature that in our manuscript, sorry, it's more like an advertise for our model is maybe you, maybe or others didn't noticed, and we actually have trained the synapses, the weight, to fit the global calcium dynamics, the correlation matrix for the global calcium dynamics. So I think this is the first model that in the field that can be trained to match the global activities of the neurons. So I very much look forward to your experiment data if you have this global dynamics of the calciums, and then we can actually now train the synapses, the weights of the synapse to fit the correlation matrix.
Right.
Yeah.
No, and we could use the connectome where we see
exactly.
the sizes of synapse. There's kind of a lot of channels they can use.
Yes, good. That's all. Yeah. So sorry, I have one additional question about how to use the C. elegans model in artificial intelligence. Have you noticed there is a group in MIT? They work on the C. elegans model for a long time, and they now recently raised up a company called Liquid AI. Hasami, and I mentioned him and his work is not only like using the simplified C. elegans for the artificial intelligence, for the autonomous drive. It's very interesting when I occasionally look at his PhD defense PPT, which he put online. Actually, in one of his page, he explicitly described a simplified circuit as a causal inference. Have you read his work or related work? And
I looked at it. I'm personally a little skeptical. And like, there's a long history of deep learning people wanting to learn from brain data about how to build better autonomous systems and so forth. The real inspiration of how neuroscience has affected neuroscience has always been pretty abstract. Now, like, there's neurons, they have nonlinear input-output functions. There's no negative spikes. There's kind of like a lot of inspiration at that, or like attention, which is actually a cognitive, not a neural phenomenon. There's a lot of that like high-level inspiration, but I've never seen anyone really get any mileage out of using the details there. Exactly. And maybe they'll teach us wrong, but my money is still on like what will really help us is more like the big, broad stroke ideas.
So what's the next question? Yeah.
Okay, I can say something about it.
Yeah.
So one thing that's very common in neural findings is that we have some dimensions. So like we take neural activities and we record from 10,000 neurons over time, we put it through principal component analysis. We will almost always find that the biggest singular value, the dimension along which most variance happens, describes an awful lot of variance. And then we have further and further components that describe less and less. And usually it looks like a power law. And the question is, and it cuts off relatively quickly. The question is what that means for encoding and what that means for decoding. So for not like encoding, decoding, no. Yeah, so for encoding, what I want to do is I want to describe future neural activity. It helps me a great deal if my signal is relatively low dimensional. Why? Because it's low dimensional, I need equations with a small number of parameters, which I can efficiently fit. And when we do things like L2 regularization, that's exactly implicitly what we do. And so for where that's for, well, that's true for both decoding and encoding, but it's basically if our signals are low dimensional, then both of these problems become easy. And signals in practice are like low dimensional in that sense. Now, let's talk about the differences between that. How do we interpret, what's the question that encoding goes with? I'm asking, how can I predict what that neuron does? What do we have in decoding? How can I combine all neurons to predict what the behavior is? If I have all the neurons, it will, of course, help me a great deal if all those neurons are correlated with one another. If I have a single neuron, I need to make do with the data of that one neuron that I have. And so therefore, I'll generally be much better at decoding because I can use all the neurons than on encoding if I can use just one. Now, what else should we say about the difference?
No, actually, I also have some puzzlings for you. It's about the BCI, the brain and computer interface. We know, and I noticed because occasionally I read some news, and we use the BCI now start to treat some brain disease. I like to read the input from the brain by inserting some probes, right? And then give some feedbacks to the brain, such as in Parkinsonian disease or treat the blindness. So sometimes I'm really puzzling. I have this puzzling question is, I think the feedback signals to the brain is my personal view is in very bad quality. It has nothing to do with the realistic and activities. But sometimes amazingly it works to some extent. So let's say if I give like a low quality inputs in the brain and it works, then why bother our brain have so complicated architectures? So that's my question. So yeah, so I really, because when I read this optogenetics paper, for example, sometimes they this is not about the BCI, but the same puzzling part is they just inhibit or the neurons or disinhibits the neurons and give a population, and then the mouse starts to move properly. And so it's a very rough control. It's not a very precise and elegant control. And then they argue that you can read many like Nature Science paper, right? They argue that then they restore this movement or blah blah blah. So so the same question is that yeah, we can somehow we can perturb or we can rescue the brain with this very rough control. You know, this feedbacks why bother our brain develop so complicated?
So let's break down that space a little. Now, like there exists one corner of the bidirectional BCI space, where say people who have Parkinson's, we stimulate them at times where it looks like they have real symptoms, more symptoms than normally. Or people who have epilepsy, we only zap their brains when it looks like they have an episode coming. Now, like there are those things. Those applications, those are very low dimensional things. Now, like they aren't like fine grained like me dancing or something. They're more like, now I'm doing badly. So detecting those is of course much easier. There's another branch, like of bidirectional BCI stuff, the high dimensional things, where say research of the late Sleem and Bensmeyer try to say, if I give you a prosthetic device, can we make it that the feeling you have in your finger kind of comes right back into your brain? Okay. That requires you to construct something much more complicated. Okay. The way I understand the status of the field is not that they're really good at producing these stimuli. It's more like, yeah, you'll know if something touches you or something. It's not that it has like this fine-grained texture discrimination. It doesn't feel quite the right thing. But for lots of real-world practical applications, you just need low-dimensional feedback. They're like, do I already touch an object? Yes, then I can start closing my hand. So giving back low-dimensional information already helps a great deal. So that's my reading of the bidirectional space. I do think that the space of my thoughts is pretty high-dimensional.
Yeah, I agree with this. But actually, I think it's like this. For example, if you talk about like the motor control, maybe we can use something like 20 of 30, 40 dimensionalities of whatever neural responses. Maybe it's good enough for control a two-dimensional movement or three-dimensional movement. But actually, I think that's because our task is too simple. First, second, I think there are there's no some kind of perturbations or some different kind of contexts or a lot of other things. If we add more noise, then you need more dimensionalities, I guess. I don't know. Yeah, I think it will be the brain has its. I guess the brain has its. The brain is efficient. The signal processing is really efficient. I don't think there are a lot of neurons that is not really working or something like that. I don't believe that. So I think it will be some kind of situation. All the things will be useful.
Yeah.
Point. Yeah.
Mm-hmm. I totally agree. Now, like when we look at data sets where we have very simple behavior, and like there's all these experiments with on the motor system where you just move the hand in 2 dimensions, and there's all these experiments that like Elon Musk's and others are using, where it just has a cursor moving on the screen. The more rich you make the behavior set, the more high-dimensional the neural activity. Yeah, exactly. So I think it's really an artifact of using boring tasks that make it look like the brain is so simple.
Yeah, I think so. Yeah.
2 questions from the online audiences. The first question is, after the neurons learn the new causal connections, how the neurons itself changes physically. And the second question is, could you please make comments on Carl Thurston's free energy principle?
Oh, wow, that is a curveball question. So Carl Thurston's free energy principle says that what the brain does, we can formulate it as the optimization of some energy. It's a well-known finding in the optimal control literature that basically any inference problem can be rewritten as a control problem, and every control problem can be rewritten as an inference problem, and that both of them can be rewritten trivially as an optimization problem. So in its core form, it's basically vacuous. Basically, yes, he discovered what the field knew many years before that we can write those prompts in a certain way. Now, Carl Thurston was very helpful in the sense that Carl Thurston focused us on active inference. It was an active field in computer science that fell somewhat out of popularity, but it basically, the fact that we choose our sensory stimulus. Now, like people are always like, okay, the visual information is going in. No, that's not true. I look at you and look at you, and my needs and my wants are reflected in how I consume information. That's a phenomenon called active inference. That's something that Carl Friston has popularized out of that. But altogether, I think the pieces that Carl Friston has are entirely standard, and it's unclear what we get out of it. The worst thing is Carl Friston's free energy principle does the same thing that standard machine learning does and uses different words for it. So anyone who has training in machine learning will have great trouble to realize that Carl Friston does what everyone else has been doing for a long time. So, a lot of neuroscientists are big fans of Carl Friston's free energy principle, but the people who are good at evaluating how much it adds to the field generally are not big fans of his approaches. So I personally am rather unclear what the free energy principle is contributing to the field. With respect to the other question, do you want to disagree? Come on. Yeah. Do you want to disagree?
No. Carl Friston visited Beijing a couple of times. Also, he gave his wonderful speech about the free energy a couple of times. And
he's a great speaker.
Yeah, and I think I also asked him like a lot of questions. And to be honest, till today, I think I'm not smart enough to understand free energy. So in general, I also make my own personal views. First, for me, because I'm a computation neuroscience, I use a bottom-up approach. You know, the guy like me always like disagree with many. I do not understand the many top-down methods. It's like, how can we bridge this top-down view with the bottom-up? I'm quite skeptical if a general principle works without mentioning any details about the brain. So this is hilarious if this works for science. So this is the first thing. And the two thing, I think one piece, this is just my personal view, okay? One piece that might missing in free energy in the whole theory is it does not include neuromodulation at all. It's only consider about excitation and inhibition and something like that. So let's say if his view is correct, but what is the role of the neuromodulation? We know a very tiny piece of this neurotransmitter like 5-HT or like the dopamine or like many other very, very small tiny piece of this neurotransmitters. They can significantly change the whole state of the brain. How could this link to his free energy view? That is
but let me push a little bit on that.
Yeah.
When I talk with people about Carl Friston,
yeah,
everyone says they're not smart enough to understand him. And if he is so smart, wouldn't he be good at explaining it? So what I believe is we should never take other people's theories as correct until they convince us of there being evidence for their theories being convincing and good.
So it's
like everyone's like, if they don't understand things, they're like, oh yeah, you must be right because I don't understand it. No, like basically, until we understand it, it's not a contribution.
Yeah. So it's a theory. So it's a theory. So anyway, so my conclusion is, I think the free energy is a great theory. It's a theory. Yes. Okay. So what is the first question?
What was the first question?
Yeah. How it changes?
It physically. Oh yeah. Do you want to take that? I think you're closer to it.
I mean, Well,
Oh, here's the microphone.
I'm not very clear about the. I mean, okay, here's the thing. First part of this is like, you know, I don't know, you know, how can we change the causality of the neuron, or how can we do that? Or I'm not sure about that part. But the second part is about something like, you know, if we change something, how the neuron firing change. Actually, I can give an example. You know, this is a really high level question. I can give an example about, you know, real kind of experimental data. So here's the thing. You know, brain-machine interface is really causal, because, you know, every movement of the for example, the cursor on the screen is coming from the neuron firing. About, you know, the neurons we record maybe 20, 30 neurons. This is really causal. And the thing is like, you know, after a kind of period of learning, the monkey, for example, we are running the experiment using monkey. The monkey will learn to use this kind of brain-machine interface system, and then eventually it can control this cursor moving, you know, exactly as what they want to do. And then this kind of, you know, relationship is built up through learning, and this kind of learning will really change the neuron firing. Actually, it will the population of the neuron, they will respond. The population response will you know, consolidate to some kind of pattern, and then this kind of pattern will be consolidated, and then you know, will be remembered some kind of, and then it can be recalled after the training. So in general, it's not something like, you know, how the neuron firing will change. The firing rate will not change. I mean, there's no conclusion about, you know, firing rate will change higher or lower or something like that. But actually the population kind of, you know, the pattern of the population level response, something like that will change. This is my answer. One example of the learning. It's really causal, and I don't know how can change the causality, whatever. I don't know.
I think I can fill in a little basic biology for people who don't come from that area. So the causal change of the influence of one neuron on another, we call that plasticity. It means that the strength of a synapse changes.
Okay.
And we know a lot about the biology there, and there's many factors to that. So the first thing that often happens is that molecules slightly change their properties. Phosphorylation, for example, is one way in which that happens. You have the neurons to communicate use neurotransmitters in their vesicles there. We will have a larger number of those as the connection gets stronger. We then have physical changes where the size of the synapses changes. And then we have physical changes where the whole connection sometimes is taken out. For weak synapses, on like, we start having multiple connections for the very strong connections between neurons. So the changes in causality basically happen in all possible ways of the mechanics of the neural hardware that's there.
Okay, I get it. So this is something like, you know, the mechanism we talk about, like, you know, credit assignment.
Yeah,
Yes.
exactly.
But
the mechanism for credit assignment.
Yes, but I have a further question about this actually. So do you think, you know, the structural you know, change will really get the functional change? Or I mean, it's let me say it this way. Structural change maybe yes or maybe not cause a functional change. Is that true or not? I mean,
I think the answer is we don't know. So there's an old thing that neuroscientists have done, which is, if you give me an image of a neuron, you tell me how many synapses and where and how big they are, can I predict the physiology of the neuron? Can I predict for which stimuli it will be active or inactive? There is a very cool paper by Srini Turaga, but the general finding of the space is extremely hard to impossible to predict what a neuron does based on just looking at the structure that we have. So this, like, going from structure to function, we certainly haven't succeeded. Scale.
I agree with this. Okay.
Yeah.
Yes. Thank you for the wonderful answers and discussion. Is there anyone?
Actually, as you mentioned before, for the global path, actually, basically, we can do the market, but actually, it's really possible. We can do the simulated from details. We know the correlation, but actually, my question is like, is there any or if we introduce that influence, we some more new findings?
So, but of course, to start with, like you do have causality, which is you put a magnet over my head, and you then randomly choose a time where you stimulate, or a day where you stimulate, and then you're like, do you feel better, Konrad, today than another day? So basically at the effect level, you're absolutely establishing causality. And that is why these so-called electroceuticals are making it into clinical reality. Now, if we're honest, do we know why zapping the brain there makes people less depressed? In reality, we're really bad doing it. So like predicting what will help. Really like, sure, we'll be looking at parts of the brain that people associate with emotions maybe, but there's a lot of very counterintuitive findings in the electroceuticals field, if we're honest. So you have causality. Now you can say, would it help to have how causality flows within the brain? Possibly, but that's the thing, like establishing the causal effect of something is relatively easy. Like you zap my brain, am I happier or not? Am I less depressed? Am I whatever the thing is that you're treating? Whereas what is the reason why Konrad feels depressed right now is a far harder question. Like it might be related to how the world is. It might be related to the thoughts I have. It might be related to my friends. It might be related to my philosophy. It might be related to what I had for lunch. Or the coffee that I had. So basically, the causal influence of something is easy, whereas the causal chain is very, very hard. So if anything, I think what I would push much more for is just do more screening, like just TMS more brain areas, see what happens if you do that. Like basically, if we look at actual treatments for humans, they don't tend to come out of detailed neural theories. They tend to people basically mucking around and seeing what happens if they do something.
Yes, so I do not know much about causal inference, but I would like to talk a little bit about the methodologies that I use. And I make a realistic simulation of the brain. Before I came back to China, I work in a group in the Human Brain Project in Europe, and our group made literally the best models for the basic ganglion system. It's available, and the code for the models are public available. I think it's only available on detailed model so far. And the other groups, they don't share their code, unfortunately.
Yeah, that is so sad.
Yeah, it's very sad. It's very sad. I definitely agree. Even you know what happened is, even for the different groups within the HBP, we do not share with each other.
That is such an embarrassment. So everyone who's listening to that, if you do experiments and if you do simulations and you're a real scientist, you've got to share. If you don't share, this is just, it's just bad behavior. It's like, I understand like 10 years ago, people were doing that, but it's no longer okay. It's just like, we have like unwritten rule. Every group of people, every society has unwritten rules. And it's just like by now, it's an unwritten rule that if you do computational work, you've got to share the code. If you do experimental work, you've got to share your experimental results. There's a lot of journals that now mandate it. And I think there's just a cultural understanding that it's just not cool to not share your data. I really get upset about that.
Yeah. So the model that developed in my previous group, and I think is the Stratum model, including a very detailed simulation for the dopamine modulation. So I think that so far is a very good model for Parkinsonian disease, because the DHS, the deep brains, the DBS, mostly, no, not mostly, and commonly used for the treatment for the Parkinsonian. Another thing I would like to add on is during the last couple of years, and we're also developing the simulation tool and algorithms to train the biophysical data model. We can train them as an artificial intelligence model and to fit to the data that's you recorded. What I want to see next is it possible that for the BCI, we can just directly use these detailed models to interpret the data, to feed the data, to interpret the
data. To just push back a little bit on the vision of that. Now, like detailed models, you take a way of simulating maybe the effect of what millions of neurons do as whatever individual one of them does. The big problem is it has so many free parameters. Now, if I have 1 million neurons, each of these neurons, maybe I need for detailed neurons 1,000 free parameters. So I need 1 billion models, and the experimentalists haven't yet measured them. So in principle, these models are super detailed and super in amazing models. It's just that for the time being, we don't have the ability to properly use those models because the experiments haven't been done yet.
Maybe we can start with like a few thousand or 10,000.
Or maybe C. elegans.
Yes. So that is my dream. Yes, I think, and Konrad is definitely correct that it is also my dream that one day we can train the network with millions of neurons, but it will take some time. But now we can train at least we successfully train like a few thousands of cells, and that's for that size, and it's okay. Yeah, if this can be, I would be very happy if this can be helpful to the BCI models that we have. Yeah, that's he mentioned the MetaWorm. Yeah, the MetaWorm project. Yeah. Yeah, it's in progress. At least the good thing is we make the code public available. That's I want to emphasize. That's it's very generous for Zhiyuan, because that work was done, I think, at least by 2 senior researchers, me and Lei and Dr. Lema, that he talked to you, and another senior researcher fully employing Zhiyuan, and 3 engineering and 2 postdocs and 2 research interns. So it takes a lot of money it's a 5 years work. It takes a lot of money. So I would mention that it's very generous for Zhiyuan to sponsor this project and to distribute to the community for free. And it's including a simulation of the fluid environment and the cybernetics and this system and the detailed neural system. It's really a very big and project. So even if it stops, the code is still available to the community.
That's the way to do it.
Yeah, I think maybe Konrad can take some of this elegance models and build your and to fit your data and hopefully it can not only contribute to neuroscience community and also to artificial intelligence. Yeah. So yes, thank you for your question. Yeah.
Thanks so much for organizing this.